In high school, Claire Fraser had felt like an outcast because it wasn't cool to be a woman taking so many science courses. After studying at Rensselaer Polytechnic Institute, where she first became interested in microbes, she applied to medical school. Rather than accepting a place at prestigious Yale, she opted for SUNY Buffalo because her boyfriend was moving to Toronto. The director of admissions at Yale was nonplussed. "Well, young lady, I hope you know what you're doing." The Toronto connection would alas prove ephemeral. In 1981, Fraser married Venter, then a young assistant professor at SUNY Buffalo. "We went to a scientific meeting for our honeymoon," she recalled, "and wrote a grant proposal there." The power of DNA analysis of microbes has been harnessed with great success in medical diagnostics. To treat an infection effectively, doctors must first identify the microbe causing it. Traditionally, the identification has required culturing the bacteria from infected tissue, a process that is maddeningly slow, particularly in cases when time is of the essence. Using a fast, simple, and more accurate DNA test to recognize the microbe, doctors can start appropriate treatment that much sooner. And recently, the same technology was pressed into service to deal with a national emergency, the hunt for the perpetrator of the anthrax outrage in the United States in the fall of 2001. By sequencing the anthrax bacteria from the first victim, Tiger investigators obtained a genetic fingerprint of the precise strain used. The hope is that this precise information on the source of the anthrax will lead eventually to the culprit. As we learn more about microbial genomes, a striking pattern is emerging. As we have seen, vertebrate evolution is a story of progressive genetic economy. Through a widening array of mechanisms for gene regulation, it has become possible to do more and more with the same genes. And even when new genes do appear, they tend to be merely variations on an existing genetic theme. Bacterial evolution, by contrast, is proving itself a saga of far more radical transformation, a dizzying process that favors the importation or generation of whole new genes, as opposed to merely tinkering with what already exists. Indeed, recombinant technology owes its very existence to the extraordinary ability of bacteria to incorporate new pieces of DNA, usually plasmids. Not surprisingly, then, microbial evolution, too, bears the footprint of dramatic gene-importing events of the past. E. coli, normally a benign inhabitant of our intestines and of petri dishes, has morphed through gene importation into a killer variant. The toxins produced by one strain that occasionally causes outbreaks of food poisoning, killing 21 people in Scotland in 1996-97, and headlines about killer burgers are attributable to massive genetic borrowing from other species. Genetic material normally moves vertically down a lineage from ancestor to descendant. So this importation of DNA from outside is known as horizontal transfer. Comparison of the genome sequence of normal E. coli to that of the pathogenic strain has revealed a shared genetic backbone, identifying both strains as members of a common species. But there are many islands of divergent DNA unique to the pathogen. Overall, the pathogen lacks 528 of the normal strain's genes and has instead a staggering 1,387 genes not present in the normal strain. In that 528 for 1,387 exchange lies the key to the transformation of one of nature's most innocuous products into a killer. Other bacterial nasties also show similar evidence of wholesale horizontal transfer. Vibrio cholerae, the agent of cholera, is unusual for a bacterium in that it has two separate chromosomes. The larger one, about 3 million base pairs in length, appears to be the microbe's original equipment, containing most of the genes essential to the functioning of the cell. The smaller one, about 1 million base pairs in length, seems to be a mosaic made up of bits and pieces of DNA imported from other species. Complex organisms, especially large ones like humans, are by design fairly inviolable gatekeepers of their own internal biochemistry. In most cases, if we don't ingest or inhale a substance, it cannot alter us profoundly. And so the biochemical processes of all vertebrates have tended over time to remain very similar. Bacteria, on the other hand, are much more exposed to the chemical vagaries of the environment. A colony may find itself suddenly awash in a noxious chemical, say a disinfectant, like household bleach. Little wonder these highly vulnerable organisms have evolved a stunning variety of chemistries. Indeed, bacterial evolution has been driven by chemical innovation, the invention of enzymes, or the retrofitting of old ones, to do new chemical tricks. One of the most fascinating and instructive instances of this evolutionary pattern occurs among bacteria whose secrets we have only recently begun to learn about. A group known collectively as the extremophiles because of its members' predilection for the most inhospitable environments. Bacteria have been found in Yellowstone hot springs. Pyrococcus furiosus thrives in boiling water and freezes to death at temperatures below 70 degrees centigrade, 158 degrees Fahrenheit. And in the superheated water of deep sea vents, where the high pressure at depth prevents the water from boiling. They have been found living in environments as acidic as concentrated sulfuric acid and acutely alkaline environments as well. Thermophila acidophilum is an all-around extremophile, withstanding, as its name suggests, both high temperatures and low pH. Some species have been discovered in rocks associated with oil deposits, converting oil and other organic material into sources of cellular energy, rather like so many tiny, sophisticated automobiles. One of these species inhabits rocks a mile or more down and dies in the presence of oxygen. Appropriately, it is named Bacillus infernus. Perhaps the most remarkable microbes discovered in recent years are the ones that subvert what was once considered a key dogma of biological science, that all energy for living processes comes ultimately from the sun. Whereas even Bacillus infernus and oil-consuming bacteria found in sedimentary rocks are connected to the organic past. The sun shone eons ago on the plants and animals whose remains are today's fossil fuels. So-called lithoautotrophs are capable of extracting the nutrients they need from rocks, created de novo by volcanoes. These rocks, granite is an example, bear no traces of organic material. They contain no vestige of the energy of sunny prehistoric days. Lithoautotrophs have to construct their own organic molecules out of these inorganic materials. They live, literally, on a diet of rock.